Good evening. And thank you for coming to our second public presentation of Science Talks. Tonight we're very fortunate to have Scott Edwards coming from the Department of OEB, Organismic and Evolutionary Biology. And in keeping with a new tradition we've just started, our introductions are not entirely serious. And in particular, one thing that is always interesting is to hear about how people got their starts. And so my understanding is that this is how Scott got his start in high school at a place in the Bronx called Wave Hill. Is that right? And he wrote his first paper, followed by hundreds more, as far as I can tell, which was Catalog for an Exhibit of Bird Wildlife Paintings at Wave Hill Environmental Center. So if you have kids, this is a good place to get them started. He was actually born in Hawaii and got hooked at birds at a very young age from bird watching, I understand. And he was an undergraduate here and graduated with great honors and went on to a PhD in Berkeley. After his stint at Wave Hill. And so today he is the Alexander Agassiz Professor of Zoology in the Museum of Comparative Zoology. And this is what he does. So I thought that was an amazing image of, and I hope he tells us today what he does with 4 million birds. But if you really want to know Scott, you should Google him, and it was there that I found out, for example, he loves pasta, and he's really into orange. And finally, I thought I would give him a shout out. He's just come out with this book, and this is the Amazon rating. So you should rush out as soon as this talk is over and buy this, right? And with no further ado, here's Scott.
Thank you for that wonderful introduction. I haven't seen Wave Hill in a long time, so we're traveling back in time. Thank you all very much for coming out. It's wonderful to be here, taking time out from your busy schedules. And what we're going to do today is basically take a tour of modern ornithology. Ornithology is a very integrative science. It's cross-cutting insofar as it borrows tools from many different disciplines and integrates them into studying a single group of animals. And so today we're going to take a wide-ranging tour, thinking a little bit about what makes birds special, what sorts of ways have they innovated over the last 100, 150 million years, and also look at some of the challenges they're facing today. I'd first like to thank my wonderful crew here. These are my members of my lab in the OEB department. You can see all their names up top. They're a fantastic group, and among all of us, we cover virtually every aspect of ornithology. We've got people such as Maud Baldwin studying the evolution of taste. She's particularly interested in how nectar-feeding birds know that they're feeding on sugary fluids. We've got folks like Cassie Stoddard studying the evolution of avian eggs. Christy Riel, who is also an undergrad here, who's interested in avian social behavior and a system called cooperative breeding. Anyway, it's a fantastic group, and I want to thank all of them. Amanda Liu here just finished her undergraduate thesis in the lab. She studied the disease pathogenesis of house finches, which I'll tell you about towards the end of the talk. Shane Campbell-Staton here and Allison Schultz are also key members of the force studying reptiles. We call them bird ancestors, and birds respectively. Now, when I came to Harvard in 2003, in typical Harvard fashion, they pulled out all the stops in terms of announcing that there was a new faculty interested in birds. And, you know, one has to wonder what their motivation was. This was the headlines that came out in the Harvard Gazette a few months after I got here. Edwards studies birds with genetic databases, not binoculars. Now, this is unacceptable. I mean, one can't call oneself an ornithologist and not have binoculars as the main tool. To be sure, our lab does focus on using modern genetic techniques to study the evolution of birds, but all of our questions almost inevitably come from the field, and I encourage my students to look at their organisms in the field and generate questions through that process. And so, for example, this is a recent field trip that we took to Mongolia last summer with some fabulous students and professors there. We were interested in some of the affinities of Mongolian birds relative to Chinese birds, and we were actually very close to the Chinese border in eastern Mongolia. It's a fantastic landscape, and it's a very inspiring trip, and we got a lot of new inspiration for new projects there. It's also the case that in the classes that I teach, they're very much field-oriented, whether we're bird watching in Mount Auburn Cemetery, or whether, such as this slide shows, we're traipsing around the Panamanian rainforest. Getting the students into the field is a high priority, and I think is very much in line with some of the priorities that Harvard is trying to push in terms of international exposure for students. So here's our crew last spring during spring break. You can always tell bird watchers because they trip over the undergrowth because they're looking up in the trees. And not only are we interested in the birds themselves, but we're interested in some of the challenges that not only the birds, but communities in Latin America are facing. And so, for example, we have lots of opportunity to interact with Panamanian scientists. This woman here is from the Smithsonian Tropical Research Institute, and she's taking us to a very famous site called Barrow, Colorado Island, which has been the site of many famous ecological studies. Now, one of my key homes here at Harvard is the Museum of Comparative Zoology. And it's a venerable old building. It was founded in 1859 by Louis Agassiz, a Swiss zoologist. And our collections have been on the 5th floor of this museum now for about 150 years. And so it's high time they moved to newer digs. And that's exactly what we'll be doing. In just a few weeks, the ornithology collection, as well as many of the other collections in the MCZ, will be moving to the basement of this building, the Northwest Labs. It's a vast space. I can tell you that when Dean Smith saw the amount of space that all these dead birds took up, he nearly fainted. But it's going to be a fabulous facility for teaching and research. And so this is the Byrd Department today. We're the 5th largest bird collection in the world, quite a distinction that relatively few undergraduates know about. We've got global representation of birds around the world. Our database is completely searchable online, and there's an active field program in many parts of the world. Very important to emphasize the teaching aspect of collections. This is a great way for students to learn about avian diversity, and this at a time when many other kinds of knowledges, such as high-performance computing, other kinds of very technical knowledges, are, some might argue, superseding the basic knowledge of biodiversity. So it's a really useful teaching resource. Here's just a picture of some of our new digs. Both these traditional voucher specimens, as they're called, as well as frozen tissues, which we do for our genetic work, will be housed in this new facility. And so it's going to be a really fabulous center for research. So what we're going to do today is sort of take a two-pronged approach. We're going to first think about the broad-term evolution of birds and what kinds of trends that we see over the long evolutionary history. Here we're going to talk about some of the amazing anatomical structures that birds have, but we're also going to talk about some of the really unique genomic structures. This is parts of birds that we don't typically see, but which are proving really interesting when we compare them in an evolutionary framework. Afterwards, we're going to move on to sort of looking at the outsides of birds, talking more about their ecology, some of the challenges they're facing from climate change, pathogens, and other sources. Now, to give you a broad overview of avian diversity, it's important to look not only at birds, but at some of their closest relatives. And it turns out that turtles and crocodilians, of all things, are turning out to be the closest living relatives of modern birds. Crocodilians, it turns out, share a number of anatomical features with birds, and so that isn't so much of a surprise. Turtles, however, have been bouncing around the vertebrate tree in many different places, and they seem to be comfortably now settled as the sister group, as we call it, to this clade here, which is called the archosaurs. Archosaurs are crocodilians plus birds. And I thought I'd show you some of their diversity by looking at some of their nests, because that really illustrates some interesting trends. And so, for example, turtles, the 300 or so species of turtles, actually, none of them actually exhibit parental care. In other words, when the young hatch, they simply swim out or crawl out of their nests. And so parental care is simply not present at the level of turtles. Crocodilians, by contrast, many of you are probably aware, do exhibit parental care. They have very broad nests which partially use the temperature of the biodegrading debris, such as leaves and other litter, to incubate the eggs. And you may know that crocodilians display what's called temperature-dependent sex determination. So depending on the temperature at which the embryos develop, that determines whether they're going to be a male or a female. Now, moving closer to the birds, it's very clear now that what we call the non-avian dinosaurs, these are an extinct group, which included, of course, the famous Tyrannosaurus rex and other kinds of dinosaurs. We know now that many of these groups exhibited very elaborate social behavior and also exhibited parental care. And so this is a spectacular fossil discovered in Mongolia that shows this squashed oviraptor actually incubating the eggs. It used to be thought that oviraptor, as its name implies, was actually eating these eggs. But it turns out that extensive study has shown that it's actually incubating them. Moving on to the living birds, it turns out that the most basal group are the flightless birds, such as ostriches, emus, and other so-called ratites. These are then followed by the chicken-like birds, such as this malleefowl. This is a type of bird called a megapode. It, too, is famous for incubating its eggs in a thermal environment that's generated either by geothermal sources, such as volcano bases, or biodegradable sources, such as rotting leaves and twigs. And then finally, we move to the most elaborate nests, those of the passerines, which include the songbirds, which are really one of the most diverse avian groups. The songbirds comprise over 5,000 species, and so a little over half of all avian diversity. Now, if we look at, if we sort of go back to the mid-1990s, this was the state of the art in terms of our understanding of avian phylogeny. Phylogeny, of course, is very important as a roadmap to understanding many aspects of avian diversity. You can recognize some of these groups here. Here are flightless birds. Here are the many intermediate groups, such as owls and water birds. Here are flamingos and pelicans. And then here are our passerine birds, again, comprising over half of all species. Now, fast forward about 20-some years, and a group of us now are working on a vast and extraordinarily ambitious project to place avian phylogeny on a more firm molecular footing. So groups from all over the world are sequencing the entirety of what we call the avian genome. This is the collection of DNA in its cells. For each species on this slide, we're talking about 1.2 to 1.4 billion letters on the chromosome. We're determining all of the letters, and we're trying to use these to understand the basis of avian diversity. And you can see some of our preliminary data here. You'll notice this suspicious wall of death. This is our colloquial name for a part of the avian tree that's turning out to be very difficult to crack. What we're learning is that avian orders have diversified extremely rapidly, almost in a burst of evolution. And one of the key questions now is, when did that burst occur? The key anchoring point there is this Cretaceous Tertiary boundary, where, as many of you probably know, the asteroid hit about 65 million years ago, caused widespread extinction. The question is, was this a driver of avian diversification, such that these orders diversified after the asteroid hit? Or was it the case that a few lineages had diversified before the asteroid and were able to survive the environmental crisis that it precipitated? So we haven't gotten the answer yet. It's probably, as typical, going to be somewhere in the middle. But as you can see, we're marshaling a huge wealth of data for this very important problem. Now, I just want to spend a couple minutes talking about what makes birds so cool. They're absolutely incredible machines that have solved many problems that mammals, such as ourselves, have not solved. And one of my favorite aspects of birds is their remarkable respiratory system. So, for example, birds have a series of air sacs, which they use to shunt air through their respiratory system. This kind of structure is completely foreign to mammals. Intriguingly, they have a trachea just like we do. They also have what's called a syrinx. This is the voice box. It's not the same as our larynx. Our larynx occurs in the throat, whereas the syrinx occurs right at the base of the trachea where it splits into two different bronchi. And as you'll see, it's really a remarkable structure that allows them to produce an extraordinary array of sounds. One of the coolest aspects of the respiratory system is that the air flows unidirectionally from the tail to the head in one direction through the lung. The lung, in fact, is stiff. It's not a expanding and contracting lung like mammals. Birds don't have a diaphragm, for example. Rather, they use these air sacs to shunt the air through in a unidirectional fashion, and as a result, their ability to extract oxygen is far more efficient than in mammals. So again, thinking about this avian syrinx, this is the sort of voice box of birds, and it's a remarkable structure, particularly because birds have the capability of blowing air through either of these two side chambers that you can see here. And what I thought I would do is briefly play you a video that will illustrate some of these trends. I think you'll probably recognize the voice relatively quickly.
A bird's voice box can also produce two different notes simultaneously. It's not high in the throat like ours, but deep in its chest. Low notes come from one side, high from the other.
Great. Now, if you're like my students in ornithology class, you're probably saying, oh, can you just play the movie a little bit longer? David Attenborough has such a nice, welcoming voice. If you'd like, I can finish the lecture like this. Now, we're noticing where I live out in Concord that it's really interesting. The cardinals in Concord begin singing in mid-February when the snow is very deep and long before they actually start to breed. This is partly because they're reacting to the increasing length of day, and one of the amazing aspects of birds is they actually have photoreceptors in their skull which are responding to the increasing day length and ultimately causing them to sing. You can see from this sonogram, again, this sort of antiphonal pattern that we hear in birdsong on either side of the trachea. So this is the little trills that you're seeing here. And what you can see is on the right and the left, you can see the pressure being measured, and you can see the airflow through the left and the right sides of the syrinx. I hear these every morning on my walk from Porter Square down to the MCZ. Now you'll see this call here. So you can get a nice sense of how birds right around here are using this remarkable structure, the syrinx. And what research at the MCZ has shown is that this elaborate system of air sacs and other aspects of the pulmonary system are present actually in some of these non-avian dinosaurs. We're learning more and more that these dinosaurs are, in many ways, the true innovators, because we see many of the key traits that we thought were specific to birds, including feathers, for example, appearing in these very basal ancestral dinosaurs. A final example of the respiratory system actually goes right to the molecular level. Here you see two species of geese, one the bar-headed goose, which is famous for its high altitude migration over the Himalayas, migrating from Central Asia to South Asia. The bar-headed goose has evolved a remarkable structure of hemoglobin, which of course is carrying oxygen from the air to the tissues. And key residues in this hemoglobin have allowed it to achieve a much higher oxygen affinity than its lowland cousin, the gray-lag goose. And so this is just another example of the adaptations that birds have, in this case extending to the molecular level. Now, let's talk a little bit more about some of these amazing adaptations at the molecular level. One of the things we've known for a while about what we call the avian genome, this of course again is the collection of genes in the bird's chromosomes, is that bird genomes are streamlined. They're about half the size of the human genome. As you can see, they have very little what we call repetitive DNA. They're sort of free of all this junk. They have these tiny little microchromosomes, which are on a genomic basis, really just dots that you would see under the microscope. They're very, very small compared to our chromosomes. Now, you might be saying, well, this makes sense. I mean, birds have to lose weight. They might as well get rid of all this extra DNA. As crazy as that sounds, there may actually be a grain of truth insofar as we're learning that avian genome structures do have some relationship to flight. So what you can see here is a comparison of genes in both mammals and birds. Genes, of course, are the information conveyors of our genome. You can see that whereas mammals have these long, stretched out genes with very long so-called introns, introns are basically these intermediate parts of genes. Chickens, by contrast, are virtually all business. Most of the gene is actually coding for a protein. Now, several years ago, researchers compared the intron sizes of humans and chickens, found that chickens had shorter introns, and made the probably premature proclamation at the time that this was because birds fly. How they went from a comparison of two species to the conclusion that they have small introns because birds fly, I don't know. But as an evolutionary biologist, I was sufficiently suspicious that I wanted to check this out for myself. And so what we've done is to undertake a much broader survey where I will confess we were looking at genetic databases. And this is what our survey showed. What you see on this slide is on a log scale intron size. This is measured in base pairs. And on the x-axis, you see a number of different mammals, including bats and birds. Now, intriguingly, we found that both birds and bats display smaller introns than their non-flying relatives. This is quite striking, and frankly, I was very surprised at it and had been somewhat equivocal before that. Now, why would small genomes be useful in the first place? Well, the suggestion was that to facilitate the very high metabolic rates that birds exhibit, they might want to shrink not their genomes per se, but their cells. If one can increase the ratio of the cell surface area to volume, that might facilitate many of the high metabolic rate activities that require lots of metabolic flux across the cell membrane. Shrinking the cells would then in turn shrink the genome itself. And in fact, there's a very good relationship between cell volume and genome size across many different animals and plants. The question is, what did it actually have to do with flight? Well, to answer this question, Chris Organ, a former postdoc, and I wanted to look at a situation where we could actually reconstruct the evolution of genome size, not only in birds, but in some of their extinct relatives. So what we did was we measured a type of cell, namely osteocytes, in these cross sections of bone. Osteocytes are sort of the gaps left in the bone matrix once the living cells have died. We wanted to correlate those cell volumes with known genome sizes from living species. That's data that we can easily get. Then we wanted to measure the cell sizes of extinct groups, such as non-avian dinosaurs and this moa. If we know the relationship between genome size and cell volume, and then we can measure the cell volumes in the osteocytes of these extinct animals, we might be able to estimate genome size through time in the lineage leading to birds. So we did a lot of measuring, and Chris went to museums all over the country to measure these osteocytes. You can actually see in this comparison, which are at the same magnification, that the osteocytes of this mammal, a bear, are visibly bigger than the osteocytes of Deinonychus, which is what we call a theropod dinosaur, a very closely related lineage to birds. Now, we were then able to establish this relationship between osteocyte volume and genome size among living species. And then we measured the osteocyte volume of the extinct dinosaurs, such as these theropods in red here, as well as ornithischians, which is the other group of dinosaurs. Despite their name suggesting birds, they're actually not the ancestors of birds. And you might already see where I'm going, because what we found was that the theropod dinosaurs had estimated genome sizes that were very, very bird-like. So what you see on this slide is, in these hot pink colors, the estimate of genome size, not only for modern birds, which are up here at the top, but for their non-avian dinosaurian ancestors as well. And you can see that, in fact, many of these extinct lineages had already achieved a small genome long before the origin of flight. So what you can see here is that the first appearance of streamlined genomes appeared way back here with dinosaurs such as Herrerasaurus, a very basal dinosaur from Argentina, as well as long before the actual first appearance of flight, which many scientists place around this clade, leading to a very well-known Chinese fossil called Confucius ornis. So this suggested to us that it wasn't flight per se that might be driving down cell volume and genome size, but rather it was perhaps the high metabolic rates that birds and their dinosaur ancestors originally possessed. Now, we're learning all kinds of other cool things when we look at the genome of various different groups. This is a figure from a study that's soon to be coming out, where our lab was able to provide resources to study the genome not of birds, but of turtles. And one of the discoveries that we found was that turtles, being another tetrapod group that has lost its teeth, turns out they have inactivated some of the same genes that birds have, who of course are also toothless. And so, for example, these genes such as amyloblastin and enamelin are both dysfunctional in turtles and in birds, but the actual mutations that debilitated those genes are actually different. And so we know that they're different evolutionary events leading to loss of teeth. That also goes for this gene down here, amelogenin. And so looking at the genome provides a really interesting window into many traits that we're comfortable with at the organismal level. Now, I want to talk about another really important innovation in birds, and that, of course, is feathers. I'd also like to thank Jeremiah Trimble, our collections manager, for taking some of these beautiful pictures. Now, feathers are a remarkable structure. They are composed largely of keratin, and so the same protein that, for example, makes up our nails or our hair, but it's a little bit of a twist. And in fact, when we look at the genome, we can see whole batteries of types of keratin that have duplicated specifically in the bird lineage. Now, this question about the origin of feathers becomes even more important because I was telling you, it's very clear now that many of the ancestors of birds also were feathered. And so in many ways, we're really interested in figuring out when this innovation arose and under what circumstances it arose. So these are just some examples. For example, Archaeopteryx, the proverbial first bird, famous series of fossils from Germany about 150 million years ago, had exquisite feathers, as you can see from these impressions. But of course, it also had a lot of reptilian features, such as teeth and a long, bony tail, which you can see here. Here's another example of a Chinese fossil. Remarkably, paleontologists have been able to not only infer the presence of feathers, but the actual color of feathers by looking at these small structures called melanosomes that are present in the fossilized feather impressions. Now, to look at the evolution of feathers at the genomic level, we're going to turn to a part of the genome that we call the dark matter of the genome. This is, of course, analogous to that 99% of the universe that we really still don't know what it's composed of. The dark matter of the genome refers to these vast stretches of chromosomes that don't appear to have any function. We can't find any genes in them. We can only dimly recognize certain features that we can find in other related genomes. And so, intriguingly, the human genome and bird genomes, to a similar extent, only have a few percent of their genome actually made up of genes. So literally about 1% to 2% of our genome is actually making protein. The other 98% is, with a little bit of exaggeration, anyone's guess. So we turn to a part of the genome that presents something of a paradox. We turn to a part of the genome that is conserved, meaning it's similar across distantly related species and therefore hasn't changed through evolution, but in fact doesn't actually make a protein. It's a paradox because we can imagine how proteins might be conserved because they have important functions. You don't want to tinker with something that's doing a good job. However, the vast dark matter of the genome, it's less clear what its function would be, and so the fact that we might find some conserved regions is really pretty striking. It tells us that this part of the genome is doing something, even if we don't know what that something is. So here you can see a screenshot showing how a region of human chromosome 10, far away from any actual coding regions that are making proteins, is nonetheless highly conserved across these species all the way to zebrafish. And so this is what we'll call a conserved non-exonic element. It's sort of a fancy word for one of these paradoxical genomic regions. And what we're going to do is we're going to trace the rise and fall of these conserved non-coding regions throughout the ancestry of birds. So what we did is we went to the literature and we actually looked at genes that we knew were specifically associated with feather development. There's actually a very rich literature on the actual genes and developmental processes that are giving rise to feathers. Remarkably, we found over 200 such genes. We then used a battery of tools from an emerging field called comparative genomics to discover these conserved regions of genomes across 14 genomes going all the way back to fish. And we were able to annotate almost 450,000 of these conserved non-coding regions. And just to demystify them a little bit, this is what they are. They're simply stretches of DNA that we can line up easily with other species, but they don't actually make a protein. Now we're going to use the principles of phylogenetics to figure out when these conserved elements arose in evolution. So for example, if we find an element that's shared between chickens and turkeys, we can reasonably speculate that it arose in the ancestor to galliforms or game birds. Similarly, with chickens and zebra finches, we might say it's a bird-specific element, or we might find an element that's shared between chickens and humans. Now, in that case, even if we haven't found that region in some of these intermediate species, it's a reasonable evolutionary prediction that that element would have arisen in what we call the amniote ancestor. That's the ancestor of birds and mammals. So through this technique, we're able to record when different elements arise in evolution. Again, this is just another example where you can see in front of this gene called S-HOX, which turns out to be very important for not only feather development, but other developmental processes in birds as well. You can see that it's surrounded by an array of these non-coding conserved elements that have arisen at different times. And so, for example, the red elements here are all what we call bird-specific. We only find them in either chicken, which is the reference for all of these comparisons, turkey, and zebrafinch. We don't find them in reptiles or mammals, for example. But we also find elements, for example, this green one here, which we can actually find a trace of in the Anolis lizard. This is a small element that we can easily align with a non-coding region in the Anolis lizard whose genome is also sequenced. And through this approach, we can begin to catalog the evolution of these elements and ask when they arose. Now, we can do that also for the genes themselves, and what I'll show you on the next slide is sort of our summary of all this analysis. What was remarkable to us is that it turns out that the actual genes that birds are using to make feathers are extremely old. They've been in the vertebrate genome. Even since fish, we can find many genes that are involved with feather development now, but which are doing other jobs in fish and reptiles and mammals, for example. And that's what this blue dot indicates. Most of the genes involved with feathers originated well before vertebrates and tetrapods began to diversify. That was perhaps not a shock for developmental biologists working on this, but no one had ever actually quantified it to see the big picture. Now, by contrast, what you can see here in the red is the sort of rise and fall of some of these non-coding elements. And what you can see is that there's been a burst of evolution in the ancestor of the birds, reptiles, and mammals, so the amniotes. But perhaps more intriguingly, there's been another burst of evolution right at the base of birds where we might expect the regulation of feather genes to be really, really changing quickly. And of course, these conserved non-coding elements their main job, we suspect, is to regulate the deployment of genes, the where and when of their expression. So, although the origin of the coding regions seems to be very ancient, the origin of the regulatory regions seems to be much more dynamic and much more innovative. And we suspect, in fact, that it's this regulatory evolution that is really what's driving feather evolution. It's not the genes themselves, but it's how they're deployed and how they're put in different combinations to make new structures. Okay, well, I know some of you are probably saying, we've had enough DNA, let's talk about the real McCoy. Let's do that. And what I'd like to do now for the second bit of the talk is to tell you a little bit about the current state of birds and to try to paint a portrait of them as a rapidly changing lineage in the face of many different environmental challenges. So I've been very fortunate to have worked some with the Massachusetts Audubon Society. And this has been a really interesting experience because the Mass Audubon is very interested in tracking the health of Massachusetts birds. It's heartening actually to say that in fact the awareness of ornithology and what we might call the state of the birds is increasing dramatically in the United States. And in fact, every year now, scientists and policymakers are making a document that we call State of the Birds, which is an assessment of how birds are doing across the country and in various different states. Now, this is a type of data that's being generated from the new breeding bird atlas that Mass Audubon will soon be producing. What you can see here is a slate of pixels for the American robin, which indicates a comparison between the last survey in the mid-70s with the surveys that have just been completed. And as you can see from this diagram, the populations of American robins in Massachusetts are very stable. The numbers are very comparable to what we saw 40 years ago in the surveys in the 1970s. Now, let's look at another species. Here's a species which has undergone dramatic gains in Massachusetts. I'm curious if anyone in the audience might have an idea about a species with this distribution, which has increased remarkably since the 40 years ago. Any thoughts? Pigeons? I think I heard turkeys. Okay, this is the wild turkey. So, you know, it's when you see turkeys like walking right through the campus of MIT, you know that there's a population outbreak. So this is a heartening story. What's a little more troubling, however, is some of the species which we see declining in these two surveys. And so American kestrels are perhaps the most marked species in terms of their decline over the last few decades. It's unclear why this species is declining, although it's likely that a combination of habitat modification and climate change may be changing them. Another example is the American black duck, often confused with mallards. They look very much like female mallards, but black ducks seem to be declining as well. Now, this is just one example of a trend that we're seeing across the country in terms of the fate of avian populations. What you can see here is a comparison of the wing lengths of songbirds that were caught in mist nets at a famous area for bird conservation in California, Point Reyes Bird Observatory. What you can see here on the y-axis is the extent of change in wing length comparing birds measured in the 70s with birds measured just a few years ago. And what you can see is across all these locations, there is a consistently positive change in length of the wing. Now, there's a lot of speculation as to what might be driving these changes, these very rapid changes in body size. Some of you may be familiar with what we call Bergmann's rule. Bergmann's rule states that individuals of a species living in very high latitudes and cold climates tend to become larger. And we see this in many, many different species and birds and many other groups. This, as you can imagine, is actually running counter to Bergmann's rule. The climate's getting bigger, and yet so are the animals. Now, full disclosure, other studies, for example, conducted here in Massachusetts, suggest the opposite trend, that avian populations are actually getting smaller as the climate is getting warmer. And so it's very unclear what's driving these trends, but it's certainly something to watch, and it's, I think, fascinating that we can measure evolutionary change on such a short time scale. Many of you, I'm sure, are familiar with the fact that birds are changing their annual schedules very much in sync with climate change. And so, for example, this top graph, which was produced by a group working here at Manomet Bird Observatory, shows a clear decreasing trend in the mean arrival date of various bird species. So each point here is a different avian species. You can see there's surveys from the 70s to upwards of 2000. And so this sort of change in what we call the phenology of bird populations is very consistent. We see it both in Europe and in the United States, and is perhaps more clearly linked with an increasingly warming climate. Now, we also see responses of birds to other kinds of recent assaults, and this is a slide from a colleague of mine in the University of Leiden, where he was measuring the sounds, the songs that birds make in urban environments, such as those in the red stars here, as well as in forested environments very close by. So these are sort of paired experiments. And he was interested to ask whether birds are changing their vocalizations as a result of the ubiquitous low-frequency noises that comes with large cities. And what you can see here are some of his results. This is, again, a sonogram where you can see the length of time in seconds and the frequency on the y-axis. You can see here an individual of this species called the great tit. It's related to our chickadees, and this is an example of its song in a forested environment in northern Europe. Okay, so that's what great tits sound like. Let's now look at the corresponding urban environment. Now, if we look at Prague, where great tits also occur, you'll hear a conspicuous difference. Now, some of that difference could be geographic variation. Birds vary over time and space. But this particular experiment has been repeated over many different cities throughout Europe, And it's very clear that there's a systematic increase in the frequency of bird calls in urban environments. So this is, I think, another way in which birds are coping in real time with the rapidly changing environment. Now, I want to document for you another type of change that some students here at Harvard and I studied, again, using the remarkable collections of the Museum of Comparative Zoology. So albatrosses are one of my favorite groups of birds. We've got students in the lab right now studying assaults such as plastic in the environment and how they impact these majestic creatures. Elaine Vo, an undergraduate who is now a graduate student at UC Berkeley, was interested in the possibility that these seabirds, despite nesting on very remote island archipelagos, might be suffering some of the consequences of heavy metals. We tend to think of islands as very pristine environments, but with all of the increasing numbers of pollutants in the atmosphere, it's clear that maybe no populations are immune. So we didn't exactly get to go to Hawaii, but what we did was we used a series of museum specimens in the Museum of Comparative Zoology, as well as specimens from other collections that were from the northwest Hawaiian Islands, where these black-footed albatrosses breed, as well as from the Japan Islands in southern Japan, where there are small colonies. And we were able to assemble a series of specimens that spanned about the last 120 years, again, using specimens that were collected with no thought of measuring heavy metals in them at all. And what Elaine was able to do was to demonstrate that, in fact, if you look at what we call organic mercury being the type of mercury that's actually incorporated into the tissues and feathers of these birds, you can find a very detectable and consistent increase over time. And that's what you see here in this black line. You can see a consistent increase in organic mercury measured over specimens over the last 120 years or so. And the levels that we're finding in some of the very recent specimens are high enough that they are probably impacting their reproductive fitness. So this is, again, an example of change in bird populations in real time. Now, I'd like to end with another interesting example of rapid evolution in the face of environmental change. And this comes from work that we've been doing for a long time now with collaborators in Alabama. Jeff Hill, in particular, is a professor there. And we've been interested in the interaction between a bacterial pathogen called a mycoplasma and these cheeky little songbirds called house finches. Many of you probably know house finches. They sing right here on Harvard campus. They're native to the western U.S., but they are introduced to the eastern U.S. about 60 years ago. Now, about in the mid-1990s, house finches started getting sick. So you could see, for example, that birds had lesions around the eyes. They had what we call conjunctivitis. They had trouble breathing, and you could see that they were very listless on the feeders that they frequent. And through a combination of what we call citizen science and science conducted in the laboratory, scientists were able to track the culprit to a bacterial pathogen called a mycoplasma. It's a very small genomed bacterium that is actually a real problem for the poultry industry. They spend millions every year trying to develop vaccines for this. And serendipitously, apparently jumped to house finch populations. Doesn't seem as if they've infected many other species of birds, but the house finches really took off, and you can see that within about 5 years, this pathogen had spread clear from its source in the mid-Atlantic states, so Maryland and Washington, D.C., clear to the Rocky Mountains, where it stalled for a few months, after which it jumped the Rockies and spread to the north and south through California and the west. So we've been trying to understand how this very recent pathogen has caused evolutionary change in house finch populations. And this is just there's a picture of the of the pathogen on the left and the house finch on the right. We've been blessed with a number of really exciting resources to study this problem. So for example, we have large series of house finch tissues collected before the disease struck in the early 1980s in the Royal Ontario Museum in Toronto. These allow us to get a snapshot of the bird before the disease ever struck. We've also done a variety of studies comparing populations such as this one in Alabama, which has been exposed to the pathogen now for a good 15 years or more, and populations in the West, such as in Arizona, which still haven't seen the pathogen. And so we're taking a historically exposed population and comparing it to a population which is still naive to the pathogen. And we've used a variety of different tools, from DNA sequencing to looking at gene expression, to study how the house finch is changing on this very rapid time scale. Now, in the next slide, I'm going to show you just a quick look at what we call gene expression. And so this is the expression of genes, some of which are involved with the immune response to this pathogen. And what you can see on these green bars, what you see here are 4 examples of genes, just looking at how their expression differs in uninfected populations. So this is birds taken from Arizona and birds taken from Alabama. We actually put them in a common environment in Alabama for about 3 months before beginning the experiment. And you can see remarkable differences in gene expression. These are telling us that the level of expression of these genes is higher in the Arizona populations than in the Alabama populations before any infections have been done. Now, when we infect the birds with the mycoplasma, we see, in fact, just the opposite. We see, in fact, that the pathogen seems to be pushing expression of these immune genes down in the Arizona population. By contrast, the Alabama population seems to be holding its own with regard to expression of these genes, as well as with regard to many of the phenotypic responses, such as the conjunctivitis that we expect to see in this mycoplasma. So this is an example of possibly evolution. It could be some environmental effects, but we are hot on the trail of some of the key culprits that are causing these expression changes. It's an example of change, very rapid change in response to an emerging pathogen in this species. And I want to end now by showing you an example of rapid change, not in space, but in time. One of our key hopes is to look at the evolution of this bird from before the pathogen here in the mid-'80s and after the pathogen has struck. And so my student, Allison Schultz, who is here tonight, has been conducting what we call a genome-wide survey of variation in the house finch, looking at birds from before the disease and after the disease. Now, our hope is to find regions of the genome that are changing between these 2 time points. And since I'm running out of time and I don't want to bore you with statistics, I just want to mention this statistic here called FST. FST is sort of a classical measure in population genetics that can tell us about the differences between different populations. And you can see how this statistic varies from 0 when there's very little difference between populations, and 1 when there's essentially complete differences between populations. And so what Allison is doing is measuring FST across the entire genome from before the disease struck to afterwards. And this is some of her preliminary results. Now, you can see here, we've looked at over 15,000 places in the genome. Now, what you can see is that FST across most of the genome is very, very small. This is boring. We were hoping for some big changes here. This means that the pre- and post-epizootic populations are pretty similar genetically. And in fact, the FST here is 0.0064, not really anything you can shout about. However, in this very, very minute tail of the distribution, which you can't even see here because our numbers here are so tall, we see a few genes which are showing really remarkable amounts of differentiation. So what you can see here is about 40 or 50 different genes which are showing FST between 0.5 and 1. This means there's been a very large, rapid amount of change, again, comparing those pre-disease and post-disease populations. This, I think, although it's preliminary, deserves a big wow. This is a really exciting result because it's rare that we find these large genetic changes between populations that are, in this case, so close in time. So I hope through all of these examples I've given you an idea of some of the amazing directions in which ornithology can take us. As you can see, we can delve deep into the genome, but we can also use a variety of clues that birds are giving us to tell us about the rapidly changing environment. And so I hope all of you take the opportunity this spring to pick up your binoculars and dust them off, get up from behind your computers, and go out and learn some of the birds in your backyard. I guarantee you won't be disappointed. So I'd like to thank the many talented people that I've been able to work with, and I'd be happy to take your questions. Yes, question.
Thank you. Yeah. Thank you. A pleasure to hear you talk about birds. For the non-specialist and for the specialist who loves birds, what would you think might be 3 or 4 initiatives that maybe, in terms of public policy or environmental policy or bird policy, that that might be the most promising for people to consider perhaps learning a little bit more about and perhaps considering being participating in, if there are any?
That's a great question. So you're interested in sort of public education about...
Well, I'm interested in how we can help birds continue to thrive.
Sure. Well, I mean, one thing we could do, for example, is have much more aggressive initiatives in urban centers to prevent window strikes. Many of you know that tall skyscrapers are death traps for migrating birds. And sad to say, even on Harvard campus, we get a fairly healthy harvest of migrating warblers every spring as a result of hitting glass windows. And so I think that's certainly not an easy thing to do, but it's a very tangible thing that could be done. Well, one can put, there's various kinds of reflective glass that are expensive but useful for deterring birds. Of course, there's the tried and true methods of putting up silhouettes of hawks, for example, or kestrels that would deter birds. And one thing that we're able to do now is to monitor very closely the migratory paths of birds, not necessarily through visual by looking at them, but actually by hearing their calls at night. And so there's an increasing number of sound detectors placed in strategic areas that are able to pick up the calls of migrating birds. And now many of these are very short chips, which most of us probably couldn't identify the species. But using various computer algorithms, we're actually able to identify the species flying over, when it's flying, and the direction it's flying. And so these are the kinds of things that I think could help us learn where birds are, where they're moving through the landscape, where we might place some of our wind turbines, for example, energy-saving wind turbines that would be in high wind areas, but preferably out of the way of migrating birds. And so, I think getting kids and students outside, if not looking at birds, then just looking at other things, I think is going to go a long way towards, increasing our passion for the natural world and, making us less obese as a species, et cetera, the whole thing. So, yeah.
A little bit related to that, I was wondering if you knew any details that you could share about, you mentioned citizen science efforts going into the mycoplasma study the house finches and what was involved in the citizens.
That's a great effort. So citizen science, of course, is a increasing trend wherein scientists mobilize the power of crowdsourcing to get folks involved with science and with discovering some really cool stuff. And so in the case of the house finches, there's actually a website at the Cornell Lab of Ornithology called, there's Project House Finch, there's Project Feeder Watch, a variety of different online tools that folks can look at and record what species they've seen. You may be familiar, for example, with eBird, which is, it's an online tool for recording essentially your bird lists. And Massachusetts is actually one of the top contributors to eBird. And it's a remarkable scientific tool because what's just a bird sighting for one person, when aggregated with millions of points, becomes a huge information source for scientists. And so citizen science, there's always concerns about the quality of the information, but there's many also effective ways to get around that. And so I think it's going to be an increasingly important source of data, particularly over large scales. I mean, we can do these experiments across the whole country, for example. Yeah.
As you look at your DNA sample, do you see evidence of crossbreeding between different subspecies of birds?
So do we see evidence of crossbreeding between different subspecies of birds? It's turning out that hybridization that you're referring to is increasingly common when we look at different bird species. And what's even more puzzling is that when we compare two different species, we'll often see that much of their DNA is separate, going on its separate way, but we'll actually find a trickle of genes that are actually moving between species. And it's turning out that this is actually a fairly common event, not only in birds, but in our own species. And so you might be familiar, for example, that about 4% of our DNA is probably from Neanderthals due to a hybridization event that took place as the human species was diversifying. So hybridization can actually be a really effective way of getting new genes and new variation that you didn't have before. So for example, different subspecies of, wolves versus domesticated dogs, turns out that there's a dark coat color, which wild wolves now have some populations that originated from domesticated dogs, and turns out to be in some circumstances actually adaptive. And so hybridization isn't so much an aberrant issue in nature, but it's turning out to be an advantageous issue as well. Yeah.
In the current National Geographic, there's a piece about resurrecting
Oh,
species.
I knew someone would ask me about that. I knew someone would ask me about de-extinction, which is this, there's a lot of hype. I'm sure all of you have seen. There was a big meeting at National Geographic, and a couple of us were helping out with a meeting about a year ago over at Harvard Medical School, where a famous geneticist named George Church is getting very excited about resurrecting the passenger pigeon, whose extinction is coming up next year. So that'll be the centennial of the passenger pigeon's extinction. Oh man, this question's almost as difficult as do I think ivory-billed woodpeckers are still flying around. Look, all I can say is that scientists are going to try it. Let's not worry about the ethics right now. I think if it seems possible and it seems like it might work, scientists are going to try it. And the passenger pigeon is one such species. The idea is to sequence the genome of a related species called a band-tailed pigeon, and then sort of systematically change those positions in the DNA that would make it a passenger pigeon. Sounds like a tall order, but I'm going to be listening with my ear to the ground. So, yep. Yeah. Yes.
With very ancient tissues, that they're concerned on whether the cell size observed are really reflected.
Right, that's a great question. So the question was, when we were doing our study of cell volume, of osteocyte volume, do we see a correlation with genome size, and is that cell volume reflective of the actual genome size? These are good questions because every tissue type has a different way of preserving in the substrate. We made extra sure that we were trying to get accurate measures of these osteocytes by making many different slices through the bone. You might imagine we might catch some osteocytes right at the top, and so they look very small. But in general, the absolute size was important but not critical. Once we established that correlation, we could then, with genome size, we could then go on to estimate its genome size in extinct species. So we think the results are robust, and in fact, Chris Organ, the guy that did that work, has actually gone on to measure other extinct species, such as pterosaurs, which are the extinct relatives of dinosaurs. And remarkably, they too have small genomes, small cell sizes. And so it seems to be a trend that is repeating over and over again. Oh, we got one more question here.
I was wondering where you sit on what you think the motivation for the innovation of flight was. I know there's several theories, so I'm just curious.
Right, flight as an innovation, that's a great question, because of course feathers, we now know, probably didn't evolve for flight, right? They were present in lots of dinosaurs that you can tell just from their anatomy didn't fly. So flight itself, you know, some people think it's a development that came from catching insects, for example, sort of a raptorial mode of life where you've got this sort of movement of the forearms. Some people suspect that, I mean, it's very clear that it's a great way to escape predators. There's still all kinds of debates about whether birds learn to fly from the trees down versus the ground up. I enjoy watching these sorts of debates from the sidelines. I don't intend to get involved with any of them. But it's a very interesting question, and who knows, maybe DNA will actually have something to tell us about that as well. So.
All right. Well, on that note, let's thank Scott for